All right, good morning, everybody. Hope you can all hear me. Okay, so we pretty much ended up here, right? Which is the idea of using a series of 2-point crosses to figure out a genetic map, right? So kind of both distance and gene order, right? So those are the two kind of main parts of a genetic map. Now, the problem is that 2-point crosses are not very efficient. Part of the reason is you have to do a buttload of them, right? So to map just to map the relative positions and distance of 3 genes, you'll need to do 1, 2, 3 crosses, right? And the more genes you do, the more crosses that you'll need to do to map that, right? So it's not a very good use of time, right? So there's a more efficient way of doing it. And also there's the issue of double crossovers. So essentially what we mean by double crossovers is this, right? So if 2 genes are far enough apart that although they're still linked, there's the possibility of 2 crossovers occurring between them. Those will not be detected in a 2-point cross. So the example here, those 2 genes are clearly still linked, right? Otherwise, you wouldn't be doing the cross. But they're not so tightly linked that 2 crossovers cannot happen between them. They'll be rare, without a doubt, right? Because of simple matters of probability. But if you do get 2 crossovers, you get recombinant chromosomes in the sense that bits of DNA have been swapped, but they're not recombinant for the 2 alleles you're looking at. And because the issue is whenever you work out recombination, when you work out map distance is recombination frequency. So if there are recombination events that you're not seeing, your map distance will be wrong, right? Essentially, you will underestimate the distance between 2 genes, right? Because again, map distance is based on what you can see, right? So you're looking for a change in allele combination, which will denote a recombination event. So if recombination events without you seeing a change in allele frequency, they're not counted, even though they are occurring, right? Even though they can, they are contributing to that map distance, they aren't counted, so they don't. Does that make sense? Yeah, essentially there are hidden events which you can't see. And so the way around that is to do a 3-point cross, right? And so hopefully 2-point crosses you're all well up on, right? Working out recombination frequency, because 3-point crosses are quite a fair bit more complicated. They're not impossible. It's just mathematical logic, again, at the end of the day, right? But there are some rules that you need to follow. And as one might expect, I have a patented plan for 3-point success. All you have to do is like those dieting plans with like milkshakes and stuff. Just do what I tell you and it'll work. Guaranteed. Okay, so let me draw this up because I prefer drawing stuff to using a PowerPoint wherever possible. Okie dokie. Come on, let's get this all set up straight. Get the full benefit of my tiny whiteboard. Right, so we're going to do a little bit of theory first, and then we'll get into the actual practice of this stuff. Because like with the chi-squared test and so on, key to success is practice. Always, always. And there will be, if my memory serves me correctly, at least 4 or 5 questions on 3-point crosses, at least in the exam, including calculating map distances, gene orders, things like that. So as you know from the last exam, you don't have a whole bunch of time to do all of this. So the better practiced you are this, the easier and quicker it will be. And just as importantly, you'll get it right. So this is actually one of the things I like about this stuff. I like teaching it because I can confidently say to my students, if you do this as I tell you, you will get the answer right. Right? It's not based on your ability to remember facts or to understand like how different things interact. It's just simple, basic math, right? So let's get on with it. So essentially what we have, and this is going to be in tiny format. I really enjoyed using my giant whiteboard at work yesterday. We have, I'm going to use pluses just because it's going to be easier for you lot to see and for me to draw. Set up like so. As you know already, you can only see recombination if an individual is heterozygous for 2 or more genes, right? So if we're doing a 3-point cross, you know that individual will be heterozygous for all 3 genes. Now again, these are homologous chromosomes. And what I'm showing you is what you have entering prophase 1 of meiosis. So these 2 blue lines are sister chromatids, 2 red lines, sister chromatids. Blue and red are homologous chromosomes. Right, just so that's crystal clear. And stop me at any point if you have any questions or you can't read stuff, right? Because this stuff is important. It's important to me that you get this. Right, so obviously, let's do it this way. Got more space. From any 2 or 3-point cross, you'll always get at least 50% of your offspring with parental, or your gametes with parental chromosomes, right? Because these ones on the outside never recombine. It's only between the 2 in the middle that recombination occurs, right? So you'll always get at least 50% parentals. Now, if you're looking at linkage, you're guaranteed that it's going to be more than 50% because linkage is what of parentals? What's the word I'm looking for? Yeah, thank you, Jacqueline and Lindsay. An excess of parentals. Exactly. Okay, so other than the parentals, you have 3 different possibilities with recombination. Right? You are going to get so this is going to be a single crossover. Hopefully you can read all this. Is that close enough? Cool, good. Always feel a little bit like, you know, size challenge when it comes to my whiteboard. Between A and B. So essentially our crossover is going to occur here, right? We're still going to get the parentals. I'm not going to show the parentals just so I get more space here, right? But you will have the parentals as well. I'm just going to show you the recombinant chromosomes, okay? just to be crystal clear about that. So something I always do, right, and you can do it too, is just use your finger to follow what's going on, right? So for the blue chromosome, you follow along till you hit the X, then you jump over to the red and follow it along. So the recombinant chromosomes are going to be plus and then BC. And because it's recombination, you always get the reciprocal or the opposite. So you go along the red, past A to the cross, cross over, literally, and then plus plus. And obviously there's also those there too. I'll just do it for that one. Just don't want you to get the wrong impression that those parentals don't exist. They do. All right. So the two recombinant chromosomes now are going to be plus BC or A plus plus. We don't have much space on here. Does that make sense so far? Again, just follow your finger all the way along until you hit, it's like literally the pedestrian crossing in a sense, right? This is where you cross the road, right? You cross over to the other chromosome and walk along the other side of the road. Sounds like my puppy's chasing cats. Ooh, this is Caramel Cat. Hello, girl. Hello, girl. Oh, she's very skittish. I'll wait till she comes back, see if I can grab her. Because the dogs are upstairs, so now the cats come downstairs. It's like mutual exclusion zones. Okay, so we also have potentially crossover between B and C. Hang on a second. Say hello. You say hello? No, she's being quiet. This is Caramel Fluffy Pants Creep. And this is our very first pet, and she is a weird, cantankerous, funny cat. Say hello. Hello. There you go. Off she goes. All right. Well, thanks, cat. Perhaps I shouldn't have brought you up on the couch. Come on, push. So the other crossover is single crossover between B and C. Right? And same deal again. For the blue one, follow it along until you hit the cross. So we're going to be plus, plus. And then you cross over to the other side and you have C. She is a very cute cat. She's really annoying though as well. She likes thrown up underneath our bed, which is really, really annoying. Although on the flip side, our puppy now goes underneath our bed and eats it. So I guess it kind of, you know, works out, I guess. Same again on the other side. Go along the red chromosome, AB, and then you cross over to the other side and you'll get plus. All right, those are the single crossovers. There are parentals in the second cross, Cynthia. I'm just not putting them in there for the sake of clarity, just because I don't have much space. You can just kind of imagine them there. You'll always get parentals. It's a good question. What the hell is that dog doing? You'll always get parentals because the two on the outside never undergo recombination. So that's why if there is no linkage between two genes, you'll get 50% parentals and 50% recombinants. It's also why if you do have linkage, you'll have an excess of parentals because you have the 50% anyway that don't undergo recombination, and then whatever percent arise from lack of recombination between those two things that you're looking at. Does that make sense? So you can never get more than 50% recombination frequency. Because unlinked, it'll always be 50-50 parental recombinant. Okay, now the added wrinkle with a 3-point cross, and this is what often trips people up, is you have your double crossovers. Hello, dog. What are you doing? Chased around by a puppy, I guess. So now you have two crossovers. One between A and B and one between B and C. And so now this is where the whole follow the finger kind of deal is really worthwhile. Go past the first gene, cross over to the red chromosome, go past the second gene, cross back to the blue chromosome. So we have plus B plus. It's kind of a shaky line because it's at a distance. And you could do the same for the other one or just know that it's going to be the opposite, right? So you'll have A plus C. All right, so now we actually get back to some of the probability and biology stuff that you learned way back when in the first section. So if we have a probability of recombination occurring a quarter of the time here, quarter of the time here, and these crossovers, these single crossovers are independent events. Whether or not you have a crossover between A and B has no effect on whether you have a crossover between B and C. What's the probability that you'll get a double crossover? They are independent events. What do you do with the probabilities for independent events? Let's say you had 2 horse races you want to bet on. Going back to my granddad who used to bet on the nags, as they say. If you bet on 2 different races, right? And so the outcome of the 2 races is independent. And the probability of your chosen horse winning each race in each horse in each race being whatever, like 1 in 10, right? Is the probability that both horses will win their races, right? So you'll win payouts for 2 races. Is that probability higher or lower than winning 1 race? Right, so is the probability of winning both races, you know, if you put your bets down, higher or lower than winning just 1 of them? It's lower, right, exactly. Thank you, Sayed and Jacqueline. Because you have to have something happen twice in your favor for you to get that outcome, not just once. So what do we do to work out the probability? We multiply it. All right, thank you, Michael. So what's the probability of getting a double crossover in this case? It's 1/16. Exactly, thank you. Good stuff. And so oh, that's all right, Jonathan. But remember, you times the numbers at the bottom, right? That will give you your fraction that you want. So that's all right. The big deal about this is that these double crossovers are very rare relative to the single crossovers, because 2 separate, potentially rare events have to happen, both of them, to see this outcome. So that's one key point. I'm going to write this down because this is like on the exam type stuff that's critical, right? And I really want you to get this. Come on, can't type. So double crossovers are the rarest. So the fewest individuals in your cross, those will be the ones that are the result of a double crossover. Also, many exclamation marks, in double crossovers, only the gene or the allele in the middle moves. Right, and so the key point here is that you can use double crossovers to determine gene order. Terrible. Camera's being tricky. Crap sticks. Give me a second, just go get fresh batteries. anybody got any questions? All right, so now we actually get into the math. And this is where stuff gets a little crazy. And another thing, and I'll write this down because it's important, even in capital letters. You cannot assume the gene order I give you is correct. You have to work it out from the data. Ah, dang it, data, that. All right, you ready? Okay, so there we go. All right. As a sign on my friend's door says, shit is going to get real. So this is the kind of information that you will be given. Make sure I've got enough space. Can you see that well enough? Do you need a light on or anything? No, I might close the blinds just to cut down on that. Okay, so let's give you some numbers. Let's just make up some shit. So let's say 1, 2, 1. All right, RB are numbers. Okay, so here is my patented plan for 3-point success. Can't remember how many steps in it. A bunch. First step. Step 1. Oh, I just remembered a Saturday Night Live skit with Adam Sandberg. Sorry, not going to repeat that because that is definitely not appropriate for class time. Anyway, what was the other guy? Singer. Curly hair. Oh, man. Anybody know who used to do stuff with Adam Sandberg? Justin Timberlake, that's it. Yeah, so they did some really, really funny skits. And it was basically something in a box. So look up Adam Sandberg and In a Box on YouTube, and you'll see what I'm talking about. So step 1, find the parentals and the double crossovers. So linkage is a what of parentals? And bang this into your heads whether you like it or not. It's an excess. Thank you, Lindsay. You might have been one of the ones that wrote it first time. All right. It's an excess of parentals. So of all of these numbers, Which ones are the highest?
The first two.
Yeah, the first two. You need to know what ones are the parentals because that will tell you which. Yes, thank you, Lindsay. That's a good point, actually. That will tell you which allele is moving in the double crossovers. All right, super, super, super important. Okay, so what are the double crossovers? Which ones are they?
The last two, plus B plus A plus C?
No.
No.
Nope, because how many of these do we have versus how many of these? All right, yeah, Miko and Amanda got it. Double crossovers are going to be the rarest. So the fewest individuals of all of these possibilities will be the result of double crossovers. Does that make sense? Good. Oops. So these are our double crossovers. These are our parentals. Two. Figure out which allele moved in the double crossovers. And sorry for the shitty writing. It's just really hard to do a stretch like that. Okay, so if these are the result of double crossovers, whichever allele has moved from the parentals is the one that is in the middle. So which allele has moved? So if one of the parentals is plus plus plus, and we have plus plus C, which allele has moved? So which is different here from here? There can only be one allele that is different. Right, it's the C, Mika. So the C has to have moved, right? Because you've gone from plus plus plus, or plus plus plus, to plus plus C. Or if you do another way around, we've gone from ABC to AB plus. So the plus has moved. Yeah? So the C has to have moved. Now you know that double crossovers, it's the allele in the middle that moves. So then you have gene order is A, C, B. Or BCA, doesn't actually matter. You have to do this first. If you don't, all the other stuff is just going to be wildly confusing. I always think of the whole tortoise versus hare deal when it comes to this stuff. You know, slow and methodical wins the race. All right. So I did it deliberately this way around because I wanted to point out or make a point that however this stuff is written is not necessarily the order which the genes are at. You have to work it out using these 3 steps from the data that you're given. You can't make any assumptions. Oh, the 3rd step? Yeah, sure, Bridget. So another way of writing it is so the middle allele, right, the one that's in between the other 2, has to be the one that's moved in the double crossover. Because it can only be the result of, let's put it up here. Right, you can only get plus C if C is in the middle. Can you see that? That's a little tiny, unfortunately. I'm gonna bring it a little bit closer. So even though it's written wrong, the numbers tell you that C is the one that moved. And so the only way you can get plus C is if the gene order is plus where it's A, C, B. So you get plus, C, plus. So really, the correct way of writing it would be plus C plus. Yeah, of course you can rewrite it, put C in the middle. Sometimes that's actually a good step to do, is just kind of rewrite these so that the gene order is now correct, right? Because that also helps simplify things a little bit. So you write it like so, right? That's exactly right, Mika. So step tells you which allele moved. Step 3 puts that allele in the middle of the gene order. So it would be ACB or BCA. Ah, you're very welcome, Bridget. So it's going to be a little bit of a mess, and I'll try and make sure that I do it correctly. So this crossover is, which one's moving here? A is moving here, right? So the only thing we need to change here is that. This obviously we need to change. And which one else? This one down here. So here the allele that's moving is B. This tells us, or this tells us B is at the end. So yeah, that was a really good point, Bridget. That's actually a very good way of doing things, particularly if you're working out with a pencil and paper. Because now it becomes really easy to see what's going on. All right, let me take a picture of that and then we'll get on to the next few steps. So this is why I like doing it out on a whiteboard. It's so much easier to explain. Well, not easy, right? Oh, Mika, you offered to, I'm not so sure about this. Add a step to Dr. Matt's patented plan. You realize you're not going to get like any royalties or anything like that from this. I don't know, maybe this should be Dr. Matt's and Mika's pasted plan, but I don't feel totally comfortable about that, to be honest. I don't know, that's making me all like, not so sure. Okay, so I'll write it down here really small. How about that? You know. There you go. Hey, Mika, I think it's a really good step. And I think it's a good step because it reduces confusion. So whether or not you do it really depends on whether or not it helps you. If it does help you, that would be awesome. Have at it. If you don't need to, you might save a little bit of time by not doing so. But again, the aim of the game here is to be the tortoise. You know, to go through this stuff methodically and get it right. Cool. Actually, I probably need to put there you go. You got to credit your sources, right? Okay. So, gonna leave all this stuff up here because it'll be a chore to write out again. Right, so now it's all pretty plain sailing, right? That's the heavy lifting of doing a 3-point cross. Pardon me. So what, oh shit, what step we're at now? 5. Now you've got to do recombination frequency between the first 2 genes. Just do it from left to right. You could do the second 2 genes if you want, doesn't matter. So that would be between A and C. Now, this is where there's a little wrinkle, right? That you have to include, otherwise you'll get the wrong answer, right? So the recombination frequency between A and C is there a number of recombinants between A and C, which will be this lot. Let's see if we get a different color. Do we? Yeah, we got purple. That's kind of it when it comes to colors, though, unfortunately. So this is A and C, right? So far, so simple. And the double crossovers. Because the double crossovers have a crossover between A and C. So to get the complete picture, the total number of crossovers between A and C are these and these. So it's the single crossovers, A and C, and the double crossovers. So that is single crossovers between A and C and double crossovers divided by the total progeny. You have to always include the double crossovers for both A and C and C and B. Okay, now I'm going to get you to add all this stuff up together. First, this is 269, no, 59. Double check my math. I'm not bad at mental arithmetic, but I'm certainly not great. Yeah, that's right. And that's 11. What's the total? Try and get the reflection. Total for, oh yeah, I mean, that's cool. Let's work out the total of progeny first. You're right, Mika, but we need both numbers. 2,280. Do I have any advances on 2,280? Not saying you're wrong, Lindsay. It's just I know that I make mistakes. It'd be good to see if anybody else has got the same number too. No need to apologize, Mika. I just wasn't being very clear about what total I was asking for. All right, everybody else get that number? 2280. Ooh, 2279. 2279. Cool. Okay. So it wouldn't have really changed an awful lot, Lindsay, but it's always good to just double check. Get the benefit of having the whole class to do it for us. But when you do these, I know for myself, the fact that I almost added this up wrong. I'm not perfect when it comes to doing math, right? So we got 2279. All of this obviously is times by 100 to get percent. And it was Mika, was it Mika 270? Crumbs, I'm running out of space. Let's do the equals over here. Nah, that's all right. As I said before, tortoise, not the hare. Although if you know this comes up in the exam, that wouldn't have made much of a difference because it would be very close to the correct answer anyway. Okay, so what's our numbers, people? 270 divided by 2279 times 100. 11.85%. And thank you, Mika and Said. So this is 11.85, or if one were to round it, it would be 11.9. So just remember the rules of rounding. And so that's percent equals map distance in centimorgans. So the distance between A and C is 11.85 or 11.9 centimorgans. All right, does that all make sense, everybody? Awesome. Cool. I think this is going pretty well. This is why I like teaching it this way, because it kind of takes a while to get through, but you bring as many people as possible with you. Okay, so. Oh, man, I shouldn't have rubbed that off. There you go. Now we have to do the same, but we do it for C and B. So now, oh, no, I don't want to use purple. wrong color. These ones down here are C and B because the only one that's changing is B. Okay, that's no problem. So just to go over again, recombination frequency. So essentially you can have two possible crossover events between, let's say, C and B in this case, right? So here's C and B in the parentals, right? It's the second and third gene, essentially. So the most frequent event that will be the result of a crossover between C and B is a single crossover, right? So the only thing that moves is going to be B. So we go from plus 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 to plus plus B, which will be the one down here. Can you see that okay? There's a bit of reflection going on. Yeah, it's coming from my side window. And then the reciprocal will be AC plus, right? This is why it's super important to work out gene order first, because otherwise all of this is super confusing. I'm just going to close my blinds on the side window. There you go. It's not gloomy now, but that's one possible crossover event between C and B. Now, in addition to that, you have a second crossover event between C and B here, right? So we've gone from C plus or CB to C plus and plus plus to plus B, right? So this is the second possibility for a crossover between C and B. The fact that there's also a crossover between A and C, you know, essentially doesn't matter in this case, right? So to be able to work out all of the crossovers, to be able to work out the recombination frequency, you have to add all crossover events between those two genes together, right? So in that case, that's going to be all of the progeny that are a result of a crossover between C and B, right? That's going to be these ones down here, and the double crossovers, right? So these, all of these individuals, these ones here, are the result of a crossover between C and B. And recombination frequency is the number of recombinants divided by the total. So that's why it's the number of recombinants that create CB difference and the number of double crossover recombinants over the total. Hope that helps. Thanks for the question. That was a good one. Sometimes it just takes explaining it in a slightly different way. Okay, anyway, so numbers, people. And it's times 100 because you want percent. Wow. Oh, yep. 127, that's that one. All right, which gives us, what's that, 138? Cool beans. So what's the distance? .06. Thank you. You did that, Saeed, without even moving your hands. That's impressive. Is that telepathic typing? Or is Saeed actually a robot? 6.06. It's super cool. I love it when people get this because It's like one of those, oh, that's how it works, right? And as soon as you see it, it's really straightforward. Okay, so we're not quite finished yet, even though you may wish we were. Because we need to do the gene order. Not gene order, done that already, gene map. Okay, so I'm gonna have to rely on you lot to remember these numbers because I gotta rub this off. This is also, again, kind of getting back to Mika's addition, is actually, if you have the time and inclination, good idea to rewrite the correct order here. Sometimes I will give you the correct order, sometimes I won't. But it does make it a little easier to access if it's rewritten. So that's actually a really good suggestion. And then the final step is gene map, right? So we know from the first step, let's put it here so I have space. Our gene order is A. Yeah, that's got to be the biggest one. ACB. That is 6.06 because I can remember. What was the other one again? 11 point something. 11.85. Yeah. And there you go. So that's how we've gone from all of this data to that. Okay, I'm going to take a picture. So now you get the choice as a class. Do you want to do a practice, or would you want to move on to the next subject, which is DNA structure? What would you prefer? Practice? Yeah, lots of pros to practice. Okay, so I'm going to give you, just going to make up another one. Let's see. I can't actually remember the exact numbers. This is 3 genes in Drosophila. And even if I could, I'm not enough of a mathematician to work out the raw numbers from that. So this is a practice. It's not kind of real life data because I didn't think far enough ahead and actually grab that off of the internet. Anyway, so what do we have? Oh, no. Now, this is going to be just trying to remember off the top of my head approximately what they are. Copy. Hang on, just trying to do it as accurate as I can. 60,000. So this is going to have to be about approximately. There you go. Try to get it approximately correct. All right. Have at it. A little bit closer. Give you 10 minutes and then we'll work through it. Or less if you get it done before then. Now imagine how many flies you would have to score in your fly linkage experiment if you wanted to do a 3-point cross. That would be a lot of flies. If you have any questions throughout the practice, just ask me and we'll work through them. Be a good opportunity to pick up any hitches or sticking points. So I got a question about working out gene order. So the first thing you have to do is figure out which ones are the parentals and which ones are the double crossovers. Parentals, these ones, right? Because you have a massive amount of them. These ones are the double crossovers because they're the rarest. Now you look at the parental gene order, or the parental gene allele combination, sorry, that's a better word. So we have, this is cis linkage because they're all the same, either plus, 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 or recessive, recessive, recessive. Now, which letter moves in the double crossovers? Down here, it's going to be Y. So this gene order is correct because the one that moves is the one that's in the middle. And so because Y is the one that's moving, Y has to be in the middle. And so this order is correct. Or it could be DMYW. It's functionally the same. It doesn't actually matter. But Y is in the middle. So it's Y, WY, DM. So that's why you need to know what the parentals and the double crossovers are, because it's the one that moves is the one that's in the middle. No problem. This is why I like giving practices, because it sometimes makes a lot more sense when someone's telling it to you than it does when you're trying to do it yourself. And so this will give you an opportunity to find out if there's a difference between the two. People need a couple more minutes. So that's a good question, Cynthia. Doesn't matter which way around you do it, but you're looking for what moves in the single crossovers. So you know these are the opposites of each other, right? So this is plus YDM and this is W plus plus, right? So you know that these are a group, and I'll typically write them together because I don't want to give you even more work than necessary. But these are a result of one crossover. These are a result of another single crossover. And so you just look at what moves. So here we've got plus plus and W. And here we have plus YDM. And so the one that's moving is that one, right? So in this case, this has moved because of a crossover between W and Y. So if you want to figure out the distance between these two, you need these and you need these. If you want to figure out the distance between the second two, you look at, find ones which have moved to create, well, as a result of a crossover between those two. And so here we have plus plus dm, so the only one that's moved has been dm. Does that make sense? So really you're looking for what is moving, because it's only going to have moved or changed from the parentals as the result of a crossover event. Okay, so while you keep working that out, so 1, 2, and 3 steps, and 4 as well, although we don't actually need to do it. Gene order is what? I've kind of already told you, but it's nice to ask. I'll give you a chance to tell me. What's the gene order? Exactly. Thank you very much, everybody. So gene order is W, Y, DM. Good stuff. Okay. Recombination frequency between Y and DM equals what? So between, oh, hang on, have I done that right? No, W and Y, sorry. W and Y. What's your work inside? Just so we can make sure everybody's on the same page. Okay, so it's 59, which is these two added together, plus these two added together, over a really big number. Times 100, thanks. I'm gonna write it real small because I don't have much space. There you go. Gives us 0.28 centimorgans. Everybody else get the same number. Cool. And that's actually, for a wide backside guess, that's actually pretty close to what it actually is. It's actually about 0.5 centimorgans. So this is going to be the same total. You're going to have to work it out once. We're going to have the same number of double crossovers. Now we need to add those together, which is what? 229. Cool, thank you. What's that give us in centimorgans? Oh, times 100. 1.02. And so then the final step, step 6, is we draw a map. Then we have Y, oh no, sorry. W, Y, DM. This is 0.28. This is 1.02. And that, my friends, is how to do a 3-point cross. Hopefully that was an epiphany for some. And actually, to be honest, it's not a bad way to spend an hour and 15 minutes in class. I much prefer doing this than just talking at you nonstop. Cool. So there are a couple, at least one, I think 2, in the PowerPoint. Oh, don't forget it's times 100, Adriana, to get percent. That's what I wrote. I know you didn't see it because I wrote it really, really, really small because I didn't have any space left. Oh, it's actually up here as well. You see? So this is both genetics class and an eye test all in one. Yeah, sorry about that. I should have put that a little bit bigger. I just forgot and ran out of space. Cool. All right, everybody. That's enough for today. I shall see you next week. Practice this stuff. It will become pretty easy second nature, right? After you've done it a few times, promise you. Cool. All right, everybody. Have a lovely day. Good weekend, and I'll see you all soon.